you ever feel lonely? We often think of ourselves as unique, independent individuals, but what if we told you that you were never really alone? In fact, billions of tiny, invisible creatures called microorganisms inhabit our bodies. Imagine that in an average adult, there are more microorganisms than there are human cells. Bacteria, archaea, fungi, and viruses can all be found on our skin, in the respiratory system, the digestive tract, or the urogenital tract, which form the microbiota. The skin microbiota, for example, contain more than 1,000 billion bacteria, which averages out to about 50 million per square centimeter, with more than 1,000 different species. The microbiota contains good microorganisms, which can protect us from infection. But there can also be bad microorganisms. An electron microscope can allow us to distinguish between different morphologies of microorganisms, but it cannot distinguish between good or bad. For example, it's impossible to distinguish between the cells of Staphylococcus epidermis and Staphylococcus aureus. In both cases, the cells have a circular shape. However, by culturing them in our laboratory, we can see that the cells of Staphylococcus epidermis are white, whereas that of aureus are yellow. Staphylococcus aureus is a pathogen, so it's important to discriminate. To fight pathogenic bacteria, we have developed antibiotics, which have saved millions of lives. However, unfortunately, there is now antibiotic resistance, which is why we need to use antibiotics wisely. What is antibiotic resistance?
This is the ability of bacteria to grow in the presence of the antibiotic, often due to genomic changes that can lead to resistance. In the laboratory, antibiotic susceptibility testing consists of seeing which antibiotic works best to slow down the growth of the bacteria. With the antibiogram technique, discs containing antibiotics are put in contact with bacteria on plate. The day after, we observe carefully around which disc bacteria were unable to grow. In this case, for instance, there is resistance to 4 antibiotics and sensitivity to the other 2 because there is a zone of growth inhibition.
How is resistance transmitted?
Antibiotic resistance can be transmitted between bacteria, even if they are very different. This electron microscopy shows how the bacteria can proceed for the transfer. By using a conjugation tube, one bacterium can transfer resistant genes to one devoid of them, facilitating the spread of resistance.
Why is there an urgent need to fight resistance?
Before 1980, there were many new antibiotics available and few resistant genes in pathogens. Today, the opposite is true. There are many resistant genes circulating and very few new antibiotics. The analysis of resistance in a pathogenic bacterium is becoming very complex. This is why, if necessary, we can extract the bacterial DNA and study it as a whole in order to understand resistance mechanisms.
And what about fungi?
Some species of yeast and mold can be infectious to humans and animals. Such infections are generally easy to cure and superficial, but in patients with a weakened immunity, they can be more serious and even fatal. And although less known than antibiotic resistance toward antifungal drugs also exists. Over the last years, new pathogens more difficult to treat have emerged. For example, the multi-drug resistant yeast Candida auris, or the mold Aspergillus fumigatus, which was put on the list of critical pathogens by the World Health Organization. Indeed, this species notably acquired resistance through the massive use of fungicides in agriculture.
What role do microbial culture collections play in the fight against antimicrobial resistance?
Collections are essential for analysis reproducibility and to support research on infectious diseases and particularly on antimicrobial resistance. Indeed, collections contribute to the fight against antibiotic resistance by providing reference strains for the analysis of antimicrobial susceptibility.
We also engage in preserving antimicrobial resistant pathogens for comparative and surveillance studies.
By integrating expertise and technologies from different culture collections, MIRRI supports the fight against antimicrobial resistance.